Hello, welcome to Biology on Miss Kestner's dining room table. If you hear someone running around, it's my cat, who is probably going to knock over my camera. But anyway, we are going to do the structure of DNA. So just a reminder, we have a cell right here with the nucleus and DNA inside. That DNA is in the form of a chromosome, so this is a chromosome. So I'm just going to zoom in on that a little bit more. So chromosome has these two halves, the short arm and the long arm. And bundled up in that chromosome is DNA in the form of chromatin. So DNA in its stringy form is called chromatin, and it's bundled up inside of a chromosome. Now we're used to seeing chromosomes in this X shape. That is because DNA gets replicated before a cell divides, so you're probably more used to seeing something like this. Just an exact replica of that chromosome. Okay, so we're talking about this DNA in the chromosome in the nucleus of a cell. We'll learn more about chromosomes when we do mitosis and cell division, but for right now I just want to give you a perspective on what we're talking about. So we're talking about the DNA bundled up inside of a chromosome. So now we're going to zoom in a little bit closer. Okay, so I'm going to show you this close-up visual of DNA. So you're used to seeing the double helix, right? You can think of it like two spiral staircases intertwined. We have one strand of DNA and then a complementary strand of DNA. Okay? DNA's full name is deoxyribose. Yep, I spelled that right. Okay, nucleic acid. And we call this shape the double helix. Okay? A helix means spiral, so there are two spirals. When we learned about the 4 macromolecules, we learned about carbs, lipids, proteins, and nucleic acids. So DNA is a type of nucleic acid. And if you remember, the monomer of a nucleic acid is a nucleotide. Okay? A nucleotide, oh, I knew this was going to happen. Come on. His name is Rocky. He is not involved in DNA structure. All right, just lay there. So, the structure of a nucleotide. Nucleotide has 3 parts. We learned these 3 parts back when we did the macromolecules, but I just want to go over it again. A nucleotide has a deoxyribose sugar. It's a pentose, a 5-carbon sugar. It has a phosphate group. And it has a nitrogenous base. Okay? There are 4 nitrogenous bases in DNA. There's more in other types of nucleic acids, but for right now we are concerned with adenine, thymine, guanine, and cytosine, often just abbreviated as A, T, G, and C. So it's always a deoxyribose sugar, there's always a phosphate, and then there's a nitrogenous base. It's going to be one of these 4. And this whole unit, this whole nucleotide, is what repeats to make a nucleic acid. So I'm going to show you kind of like a a small scale model here. So you have one nucleotide, sugar, phosphate, nitrogen base, I'll pick A. Sugar, phosphate, nitrogen base, I'll pick T. And sugar, phosphate, nitrogen base, I'll pick C. Okay? Now, in your genome, in your DNA code, the order of the letter matters, the orders of the letters. If this A somehow got switched to a T, that would be a mutation. But for our purposes right now, we're just doing any random assortment of letters. In reality, that order really, really matters. And of course, this would repeat along the entire strand. So you could have C, G, A, T, C, G, A, T, C, T, T, G, A, C, A, T, C. Okay, and it's going to keep repeating. Now, this is a double helix. So we only have one strand done so far. There is a complementary strand on the other side. How do we know what nitrogenous base matches on the other side? Well, there's a scientist by the name of Erwin Chargaff who came up with Chargaff's rule after lots of studies. He determined that adenine is always equal to thymine. Adenine binds to thymine, and cytosine is equal to guanine. C binds with G. So if we have an A here, we know that a T will be on the other side. So sugar, phosphate, T. Phosphate, sugar, A. And I know that because I had a T, and T goes with A. And then sugar, phosphate, G. And again, that's because there's a C there. So you have this base pairing rule. A goes with T, C goes with G. I like to remember that the letters with curved lines go together, and the letters made with straight lines go together. Now, something has to hold together these nitrogenous bases. Something has to hold together the two strands of DNA. It's kind of like, imagine a ladder. This would be the sides of the ladder, but then the rungs of the ladder need some kind of connection. And that comes in the form of hydrogen bonds. Now, if you remember, we learned about hydrogen bonds back when we did water, right? We did delta negative makes a hydrogen bond with delta positive. In this case, DNA, the two strands, the two helices, are held together by hydrogen bonds, and the hydrogen bonds form between the nitrogenous bases, the A's, T's, C's, and G's. Between A and T, there are 2 hydrogen bonds, and between G and C, there are 3 hydrogen bonds. And that just has to do with the number of available oxygens and hydrogens for the bonding. We'll get into the structure of each of these in just a little bit. All right, so that's the basic structure. Repeating nucleotides with all 3 components, the sugar, the phosphate, the nitrogen base, and the nitrogen bases are held together by hydrogen bonds. One more thing. You also need a bond between the nucleotides. So each nucleotide is also bonded covalently. There's a covalent bond right here and right here, as well as on this side. It forms between the backbone. The backbone is the sugar and the phosphate. So again, it's like the sides of the ladder. The rungs of the ladder would be the nitrogenous bases, but the backbone is made up of the sugar phosphate, and the bond that holds them together is a covalent bond. Okay, the specific name of that covalent bond, I ran out of room, I'll just put it right here. The specific name is a phosphodiester bond. Okay, so hydrogen bonds hold together the middle, covalent bonds hold together the backbone. So that's the basic structure. So now we're going to get into some of the nuanced details. So I'm just going to erase this. Let me make sure I have everything. Yeah, okay, we're good. So now we're going to go into a little bit more detail about the structure. This is the whiteboard I used to play school with when I was like 5 years old. So now it's actually being used for school. Pretty wild. All right, anyway. So again, I'm just going to redo the structure of a nucleotide over here. Sugar, phosphate, and of course, a phosphate isn't a circle. It's a phosphorus with 3 oxygen atoms, but you know, we're using this to represent as well as the sugar. The sugar is not a pentagon, it is a ring of carbons. And the nitrogenous base is obviously not a rectangle, it's rings of nitrogen. Okay, so first I'm going to go into the sugar. Just a little bit more zoomed in. Let me just pull up. I want to get it right. Deoxyribose sugar. I want to make sure I have it correct. Okay. So the deoxyribose sugar in a little bit more detail. It is a pentose with an oxygen. And remember that these rings represent carbon atoms. So there's a carbon here, a carbon here, a carbon here, a carbon here. And then there's actually another carbon group that sticks up out here with an OH group. And of course there's hydrogens sticking off everywhere too. There's 2 hydrogens here, hydrogens here. There's actually another hydroxide here and another hydroxide here. Okay, and then hydrogens fill in all of the other blanks. Now, a convention of organic chemistry is that carbons get numbered, and it goes clockwise. So this is carbon number 1, this is carbon number 2, this is carbon number 3, number 4, number 5. And they often get little prime dash marks next to them. You might be familiar with the prime notation from a math class. I think it's used in geometry. It's used to get here. 2 prime, 3 prime, 4 prime, 5 prime. And this is going to become important when we talk about a little detail about DNA. So just know that those carbons are numbered. And then I'm also going to show you a little bit more about each nitrogenous base. I'm not going to draw each one. I'm just going to show you an example of each. So the nitrogenous bases are split up into 2 groups, the purines and the pyrimidines. The purines are A and G. The pyrimidines are C and T. So a purine always binds with a pyrimidine. A goes with T, G goes with C. Okay? I'm just going to show you an example of each. So adenine has this structure. You are not expected to memorize this. I have to refer to a drawing to know what it looks like. So there's no way you need to memorize this. But you do need to know that it is 2 rings of carbon for the purines. 2 rings of carbon. Hold on, I need to make a pentagon here. Rings of carbon and nitrogen. And this signifies a double bond, okay? But regardless, what you want to know is that a purine has 2 rings, and this is adenine, by the way, and then a pyrimidine, such as cytosine, only has 1 ring. Okay? And I'm just giving you an example. So this is adenine, this is cytosine. Thymine would look a little bit different, guanine would look a little bit different, but regardless, purines have 2 rings, pyrimidines have 1 ring. So that's what makes them a little bit different. All right, so I'm going to show you how new nucleotides are added to a growing chain of DNA, because your cells always need to make new DNA, but they need to make it exactly the same way every time. So I'm going to show you a little bit about how the nucleotides are put together when that DNA chain is growing. So I'm going to start off with my sugar, and I'm going to show you a little bit more detail than I did on the previous picture. I am going to add my five prime carbon. This is 4 prime, and then this is 3 prime carbon with that OH group on the bottom. Okay, the other carbons aren't all that essential. It's the 5 prime and the 3 prime that become really important in DNA. And then I'm going to pick any random one. I'm going to pick adenine. So when a new nucleotide gets attached to this, when a new covalent bond needs to be made, that phosphodiester bond, a new nucleotide has to be added to the 3 prime end. So the new nucleotide has to bind, oh, I forgot the phosphate. The phosphate winds up getting bound here. Hold on, sorry. I want to just double check that I have that right. Yeah, that OH group goes away and you have the phosphate group there. So the 5 prime end is always the one attached to the phosphate. The 3 prime end has that hydroxyl group hanging off the end. So when a new nucleotide is coming in to make that covalent bond between the backbone, it has to attach to the 3 prime end. Okay, so what winds up happening is computer is doing something very strange. It's telling me my printer's out of ink. However, I'm not trying to print anything. Okay, now it went away. Sorry. So what winds up happening is a dehydration synthesis reaction occurs between the 2 OH groups. So remember, let's say you have a new nucleotide coming in with the same setup. There's that 5 prime carbon with the phosphate, okay, 5 prime, and there's hydrogens attached here as well. And then you have your 3 prime carbon right here. And what needs to happen is a dehydration synthesis reaction occurs. Let me put in the 2 hydrogens. A dehydration synthesis reaction occurs where this nucleotide binds right here. This is the setup needed, okay? You can't have a nucleotide coming in this way to attach to the existing 5' end. It needs to attach to the existing 3' end. So what winds up happening is this OH goes away, and the covalent bond forms between the phosphate group and that 3' carbon, okay? And same thing if another one were to come in. We have our 5' carbon here with the phosphate. You can always tell it's the 5' end if there's the phosphate there, and you can always tell it's the 3' carbon here with the OH. And again, same thing would occur. The dehydration synthesis takes out the water so that this new bond can form. And that is a phosphodiester bond, this one right here, okay? That is what holds the backbone together. So we say that this DNA strand is running in the 5' to 3' direction, because the topmost end here is 5', the bottommost end here is 3'. Okay? Now, this is only one strand, right? The thing is that the other side, the complementary side of DNA, is anti-parallel. It runs 3' to 5'. And this is because when the nucleotide is coming in, it can't just flip on its side. It actually, to be able to form the bond, it needs to be oriented upside down. I know it's hard to visualize. But what happens is, so we'd have our 2 hydrogen bonds binded to a thymine here, and then that thymine is bound to the sugar. Oh, no, it would be upside down. Let me do this right. I need to draw a pentagon upside down. Okay. This would be the 3' end. And then you'd have your five prime. That's 4'. This would be 5' with the phosphate. So this one is 3' to 5'. And again, this is because of the way it needs to orient itself. You can't just flip this like this. You need to turn it like this. It's much easier to see when you have a physical model. Drawing it, you should just be able to have it the upside, turn the other way. But if you actually have physical models, it would be easier to see, which was supposed to be our lesson for today. But of course, we are not in class, so this is the best improvisation you're going to get. This one, of course, would be your 3 hydrogen bonds binded to cytosine with the same configuration. Wow, this is hard to draw upside down. Okay. 5' would be bound to that 3' carbon. There was an OH there, but it got taken out for the dehydration synthesis. So you have that phosphodiester bond between the 3' and the phosphate, and then 4', 5' phosphate. Okay? And then one more. This would be a G. OH, but that gets taken away because that's a dehydration synthesis reaction. Phosphodiester bond, 3', 4', 5', and a phosphate. So now this one is running 5' to 3'. This one's running 3' to 5'. Right? This one has the phosphate sticking out up here. This one has the phosphate sticking out up here. 3' with the hydroxyl, 3' with the hydroxyl. So we call the structure of DNA anti-parallel. parallel. You can think of it like a two-lane road. On a two-lane road, the cars on the right side of the road are going this way, but the cars on this side are going this way. It's anti-parallel. They're not running in the same direction. They actually run, one runs upside down. You can think of it that way. Okay? So this is everything about the structure of DNA. Couple new vocab words. You have chromosome, chromatin, deoxyribonucleic acid, double helix, and then all the parts of the structure. The nucleotide with its 3 parts, the purines versus the pyrimidines, and then the actual structure, that anti-parallel structure with the 5' and the 3' carbons. It does take a little bit of practice, so what I'm going to have you do as your assignment, you're going to answer a couple questions, and then I'm going to have you draw out your own DNA molecule. You really understand the complexities when you have to actually draw it out for yourself. This is essentially organic chemistry. You're working with carbon-based molecules, carbon-based organic molecules. So drawing it out really does help. If you want to take it the extra step further, I would recommend actually cutting out pieces of paper. I can post an optional assignment if you want to get extra practice actually seeing a physical model. I can post that if you want the extra practice. But otherwise, this is your essential knowledge for the structure of DNA. Thanks for tuning in to my dining room table chat. And I'll see you hopefully in the near future.